Hello guys, welcome to Bacteria Brothers, and I am Eric, the founder of Bacteria Brothers. You may think that Bacteria Brothers sounds so similar to Amoeba Sisters, right? Well, you have the right idea. I got this idea when I learned amoebas and bacteria. I found it's very interesting to have the name. That is why I created this channel, well, partially because of my biology project. We'll look at the bacteria, which organelle it's most similar to. Yes, it's a chloroplast, and when you think of the chloroplast, you think of photosynthesis. That's right. That is my first video, so I'm going to make you a video about photosynthesis, which I think is the most well-known topic that I learned in biology. And this video is aiming for a quick review before the test. Wait, what? AP exams were for a month? Well, hope it still helps. So why this is a quick review and not a full-length video? Because I'm doing a biology project and don't have enough time for it. And second, I don't have the ability to explain all the concepts crystal clear. I'm not a teacher. So let's get into the topic. To learn photosynthesis, you must know the equation of it, which is 6 carbon dioxide plus 6 water molecules plus sunlight CO2 glucose. Actually, not quite glucose. It is G3P and 6 oxygen molecules. And the full equation is 6 carbon dioxide plus 12 water molecules, and sunlight yields to G3P, 6 oxygen molecules, and 6 water molecules coming out of it. And after knowing the general equation, we know that photosynthesis is being split into two parts, which is light-dependent reactions and lead-independent reactions. For light-dependent reactions, any light, just as the word dependent means. Light-dependent is also known as light reaction. They just make ATP and ATPH for the Calvin cycle. And this is the equation. So, in the light-dependent reaction, just the reaction to use light as an energy source to make ATP and NADPH to activate the Calvin cycle. You see here in the picture, light and water come into the thylakoid, and ADP and NADPH generated to the Calvin cycle. And then the photosynthesis itself is a cycle. The Calvin cycle is a cycle itself. The used NADPH and ATP, which are turned back to NADP plus and ADP, they go back to the light reactions, and the light shine on the photosystem again to generate more ADP and ADPH to the Calvin cycle. And the purpose of this is to make some sugar, which is glucose. Thylakoid contains photosystems, and photosystems is the place you make your high energy molecules, ADP and NADPH. So this is the basic picture explaining how the foundation of photosystem works, and well, this is similar to the ancient plants. So in the ancient plants, they only have just this photosystem, and look at the picture here, has the electron on the photosystem, and this electron is at the ground state, which is at the low energy level. When photon, which is a piece of light, hits the photosystem, this electron becomes excited. Oh, it jumps up very excited, right? Excited, it goes so high up and it comes down. Notice that in the picture, the heat is still shown in the picture. Why? Because all the reaction lost some energy. Yes, and it is causing entropy in the universe, which is, you're going to learn about this topic later. So basically, for the ancient plant, this is not enough, right? They cannot depend to survive, so they evolve, and we have another photosystem being added. as this picture. This is photosystem 2 and photosystem 1, and if you ask which happens first, photosystem 2 happens first, and then photosystem 1. I mean, wait, what? Photosystem 2 happens first and photosystem 1 happens later? Well, because the scientists first discovered photosystem 1, so they named it photosystem 1. That's all. Nothing special about this. So basically this is the same process. The electron is at its ground state, the photon just boom hit it and it's excited, yahoo! It goes up through, and then what is different because it used some of the energy to build the electron transport chain, called it EDC. We use that chain to generate some ADP and then down to the photosystem 1. So in the photosystem 1, they just do the same process. Another photon comes up, hit it, boom, whoa, excited and going up to make the high energy molecules in ADPH. You wonder why you need to bounce it up again, why you just don't make ADPH straightforward, because ADPH is a high energy molecule itself, so the electron must be high energy, yes, but being excited stage in order to make ADPH. And that's a simple explanation of the light reaction, and this is easy, right? Yes, bio is very interesting, and knowing the foundations of this is very cool. I mean, the next time you pass over some grass or trees, and leaves, and you say, oh, it's so interesting. This is, yeah. So now let's get more deeper, and this is probably the longest word in the biology you learned this year. According to your curriculum, photophosphorylation. Phosphorylation, I think you know the meaning of phosphorylation, just the addition of an extra phosphate group. And photo just means light. So using light and the phosphate group is just what basically the word means. So this is the overall view of the pictures. And wow, you may think this picture is complicated, but it's not. It's just what we've been covered in the previous slides. This is photosystem 2 here. This is electron transport chain here. This is photosystem 1. This is ADPH. You may wonder where is ADP. Well, we change ADP to here, and later I will explain the full process. So here is, we develop this into segments. This is the full process we just covered before. And in this process, yes, light reaction is where oxygen is produced. It's not light independent reaction. So light dependent reaction is where we make oxygen, since we split water. When we split water, this is the process called hydrolysis, which means you split water. Yes. And when you split water, you get oxygen molecules, which is non-polar. It diffuses easily. And you have hydrogen molecules. You may wonder, well, why we want so many hydrogen molecules? Because we want to make ADP. And if you guys already learned cellular respiration and in electron transport chain, you know that we just pump the hydrogen ions in one gradient, which is make this part very high concentration. So when this is in high concentration, they want to go diffuse out to lower concentrations. That's when they go through ADP synthase to create some ADP, and that's the same purpose here. But unlike cellular respiration, whose goal is to make ADP, we definitely make less ADP here. So follow the process through, the light shine on the photosystem, the electron being excited, go through the electron transport chain through this molecule and push hydrogen ions into here, all inside the cell to have very high concentration in order to make ADP. So when it have enough concentration, they all want to diffuse out, so that's why they go through ADP synthase. And how ADP synthase works is when hydrogen ions go through here, It just spins. When it spins, it creates energy, and that's how ADP is made. Think of the spinning top, which you use a rope and you slap it, then the spinning top just spins. That's what the energy comes from. When the hydrogen ions just go through, it's like a spinning top. When you slap it, it spins, and this is how you make ADP. And the process just keeps follows. Another photon hits the photosystem 1, and ADPH is made. So now we're having two materials in ADPH and ADP, and we're ready for the next step, Calvin cycle. So the steps of Calvin cycles include carbon fixation, reduction, release of GCP, and regeneration of RUBP. So carbon fixation, just addition of carbon dioxide molecules. That's it. That's how I heard about this. Fixation is not it means you really fix something. What's wrong about the Calvin cycle? No, it's just addition of carbon dioxide. And yes, it's talking about release of G3P. Actually, G3P is not same as glucose, because G3P, it can make glucose, but it can also make other molecules. And in Calvin cycle, we actually made G3P, not what we call glucose. So this is a full cycle of Calvin cycle. And let's see it in detail. So first, we just insert carbon dioxide in. And the ending process of this is RUBP, which is 5 carbon molecules. And carbon dioxide, CO2, which means have 2 oxygen molecules and 1 carbon molecule, which is only 1 carbon molecule. 5 carbon molecules, 5. 1 carbon molecules, 1, 6. When they combine, boom, they split into 3. 2. Yes. They split into 3 carbon molecules. And things that 3 carbon molecules. 3 carbon molecules? G3P has 3 carbon molecules. No, this is not as same as G3P. This is Rubisco. So how do we turn Rubisco to G3P? Well, it chemically looks the same. So how do we change it? Well, this time we need some energy. And that is when the ADP made from the previous step, the light dependent reactions at ADPH comes into use. It has 6 ADP and 6 ADPH. They come through the cycle. Boom! They release their phosphate. ADP release 1 phosphate molecule becoming ADP, adding some energy. ADPH, they release a high energy electron, and then second step reduction, we form G3P. Notice that in this picture we found 6 G3P. Wow, 6 G3P. I can make 3 glucose out of 6 G3P. No, only 1 G3P left the cycle. Because when you learn economics, I think some of you did by watching my, except for bio, we have short-term benefits and long-term benefits, and we need to consider about long-term benefits. If we make just 3 glucose, the cycle is completely down here, and we cannot restart the cycle, and that is not long-term beneficial for the plants. Plants do survive for many years, and most of the plants have longer lifespans than humans, so long-term benefit is important to them. So for the continuation of the cycle, only 1 GCP left. And if you notice what I mentioned before, there's 6 molecules of GCP, as only 3 glucose will be made. That means only 2 GCP can make 1 glucose. And for 1 glucose to be made, the Calvin cycle must be repeated twice to make 1 glucose. Remember that, very important. It will be on the test. The Calvin cycle repeat twice to make 1 glucose. And now we have 5 GCP, the cycle continues. And we're going to reform RUBP to the start of the cycle. Well, how do we do that? Well, more ADP is needed. So additional 3 ADP come here, release another phosphate, and give some energy, reform RUBP again. Wait, you may ask, how 3 carbon molecules became 5 carbon molecules? Well, if you look at that, we have 5 G3Ps, which means 5 times 3 carbon molecules give us 15. And now we have 5 carbon molecules, but only 3 of this. 3 times 5 and 5 times 3 is both 15. So that's why we came to the 5 carbon molecules, RUBP. And the cycle repeats again. Now only the last part of knowledge is in the last part of the chapter. We have C3 plants, C4 plants, and CAM plants, which CM plants. The C3 plants is just the normal plants we see, most of them. Rice, some of the majority of the flowers, yes. C3 plants, they just act like normal plants. They have breathing holes on their leaves called stomata or stoma. They breathe on the day, and they just do the process I mentioned before. And what is the difference between C4 plants and CM plants? As the description here says, the first fixed carbon dioxide near 4 carbon compounds that provide carbon dioxide to the Kelvin cycle even when stomata is closed on hot dry days. Notice that hot dry days plants want to survive. So that's hot dry days that you may think of some desert like Sahara Desert. Yes, that's like cactus. Some of the plants in the desert in the extreme areas, they evolved to have C4 plants and CAM plants for their survival. So in C4 plants, we see they have a mesophyll cell and a bundle shelf cell, and C3 plant only have mesophyll cell, and CAM plants only have mesophyll cell, and on C3 plant we only have mesophyll cell, and this is doing carbon cycle during the day, and when the weather is really hot, they still open their stomatas to breathe in the air. But notice that when you open up your stomatas, like when I open up my mouth during the very hot sunny day, I'm breathing air, yes, but like when I open my mouth, I'm also losing water inside my body. The water will be soon lost in the plant, and plants don't want to have that. They cannot drink water like humans do. So we have 2 different ways. So for the C4 plants, they use, just as they say, they turn carbon dioxide into a 4C compound. And notice that what's special about this, the 4C compound, it actually is a solid, it solidifies. So when this is a solid inside the plants don't need to open up their breathing holes, their stomata to breathe, because carbon dioxide is a gas and this is a solid. They can still close up their stomatas and let the 4C compound to transfer into carbon dioxide to start the Kelvin cycle. And for CM plant, it's also smart. It absorbs carbon dioxide at night. It saves it to the next day. So on the daytime when light is available, when light-dependent reaction is available, so they just use the same carbon dioxide to do the Kelvin cycle. Yeah, and the C4 plant, one of C4 plant example will be sugarcane, one of the CM plant will be pineapple, and this is all about photosynthesis, and I hope you have a good grade on it. And thank you for Bacteria Brothers. And last but not least, you may wonder, will this channel continues to update? Well, do you really hope that the low quality channel like this will update? It depends on the time. I'll be very busy next year taking like 5 to 6 APs. Probably don't have enough time for this, but if you love this video, I will try to upload some other familiar topics in biology, and I know this don't have any popularity, only have a few views, but if my video helped any one of you watching this, even one person, and I feel the time I made this video is worth it. I noticed that this is one video from beginning to the end, that you know how hard this is for AP student to finish their AP biology because they forgot all their knowledge. They have to learn it back in, and this is a great experience learning again some knowledge with you guys. And thank you. Bye.